Hello, everyone, and welcome to today's webinar titled Investigating Protective T-Cell Immunity Against Infectious Diseases Using OncoPEPT Platform Technology. My name is Vasumati Khare, and I'll be the host for today's session. Today's webinar will run approximately for an hour and will include a Q&A session at the end of the presentation. Feel free to submit your questions using the Q&A chat box in the bottom right-hand corner of the screen, and we will take questions at the end of the seminar. Any remaining unanswered questions will be answered via email. Please note that today's webinar will also be recorded and it will be available for viewing after the live event. Now, I would like to introduce today's presenter, Dr. Amitabh Chaudhary. Dr. Amitabh Chaudhary is the VP of R&D at MedGenome. He brings over 2 decades of experience in developing new products and bioinformatic solutions for cancer immunotherapy and vaccine discovery. He has developed OncoPEPT, which is an integrated genomics and cell assay platform to accelerate discovery of targets in human diseases. The platform has been used to assess CD8 T-cell targeted immunity in infectious diseases, which is the focus of today's webinar. We should, without further ado, I will like to hand over the presentation over to Dr. Chaudhary. Welcome, Dr. Chaudhary, and you can take the presentation over.
Thank you, Vasu. Hello, everyone. Thank you for joining the webinar. I know we are going through a new normal because of the COVID-19 pandemic, and the whole scientific community has unleashed all their technologies to help solve this problem. So in today's presentation, I'll be talking about technologies that we have used in identifying T-cell targets and how we have used the platform in SARS-CoV-2 to identify some interesting facets of SARS-CoV-2 infection. Let's see. This is the outline of my presentation. So I'll have briefly talk about the emergence of new infectious diseases and how we develop protective immunity. And this is just to form, the basis for the rest of my talk. And then I'll give you a, vignette of what are the breakthrough technologies that have given us a handle to understand not only immunity, but also identify targets to modulate the immune response that we generate against any foreign pathogen or cancer or any human diseases. And then I'll tell you a little bit about the OncoPEPT platform technology, where we have combined some of these breakthrough technologies in order to create an integrated pipeline for discovering novel targets affecting T-cell and T-cell biology. And then at the end, I'll tell you how we have used this platform to identify T-cell immunity against SARS-CoV-2. So this was a very revealing article that came out in Cell from Dr. Morens and Dr. Fauci. And this table actually tells us that emerging diseases, infectious diseases, have always, occurred from times, immemorial, like from the BC. And now there has been an increase in some of these new infectious diseases, mainly because of human activities. And over the years, as you can see from the table, hundreds of millions of people have died, starting from the plague of Athens. And even with, for example, the HIV/AIDS have taken almost like 37 million lives. So these emerging diseases do actually affect human lives at a global scale. So they are very scary. And I found this in a few lines at the end of the paper to be very revealing and kind of scary, because it tells us that the human activities that we are so aggressively pursuing and causing an imbalance with our interactions in nature, they are actually provoking the emergence of new diseases. So therefore, it's like a wake-up call for the whole scientific community to come up with a plan of how we should deal with these emerging diseases. So this is a cartoon of how we actually fight infectious diseases. So here I have only focused on adaptive immune response. There is a completely different system, which is the innate system that are the first responders when we are infected by organism. But I'm not going to go into that area because the focus of today's talk is T cell epitopes. So three different cell types actually help us to and protect us from infectious agents. The CD8 T cells. These CD8 T cells, they're activated by small peptides which are presented by class 1 HLA, and their main function is to create a pro-inflammatory microenvironment and also generate what we call a contact based killing of cells, which is its cytolytic activity. And these two together protects us against viruses. It eliminates virus infected cells. So that's the function of CD8 T cells. Now the CD4 T cell is another subset of T cells, and these T cells bind class 2 epitopes. Sorry, it should be class 2 epitopes, which are 15 to 17 mar. They are presented by MHC class 2, and their main function is to regulate the immune response, modulate the response, bring immune homeostasis, and then provide a very strong helper function to B cells so that the B cells can make antibodies. Now the B cells with its B cell receptor, they bind the viral epitopes or any epitope on any protein or non-protein material. And these B cells, when they get helper function from CD4 T cells, they actually produce high affinity antibodies. Now together the T cells are also responsible for creating adverse immune response, and together with the B cells, they also provide protection against infection and our long-term protection. So it's like a double-edged sword that if you don't modulate the activities of the cells, they can actually be harmful. So now in the next section, I'll talk about the breakthrough technologies that have helped us in discovering T cell epitopes and targets which are in the T cell space. Now, there are many technological advances that have happened in the last 4 or 5 years, but I've just focused on a few technologies that are more relevant to this presentation. The first is the massively parallel sequencing technology that has come and that has really grown in the last maybe 10 years. And these sequencing technology the cost of sequencing has decreased significantly, and therefore it allows us to sequence DNA, RNA, any material, and that becomes very relevant in the whole immune space, because now we can look at immune cells and identify immune receptors, which are the key molecules that regulate the immune response. The second is the mass spec. The mass spec technology was there for a long time, but in the last few years, the sensitivity of the technology has greatly increased, and that has permitted identification of epitopes or peptides that are presented on the cell surface. And then, from 2014 onwards, the TCR sequencing platform developed because of the primers that people had generated in order to amplify the TCRs and BCRs, and a host of analysis tools in order to understand the TCR clonality and the and its dynamics during in fact during a antigen-induced activation process. And then finally, in the last 3 years, we see a huge explosion in single-cell sequencing. And the single-cell sequencing platform has captured individual T cells and B cells and their function in a disease state or in vitro assays. And we have actually used many of these technologies except mass spec in order to build our platform. And I'll just go through it through very quickly. So our lab in Foster City, provides NGS services, and we have very high throughput, scalable sequencing platforms and analysis tools, and we sequence many different sample types, and we do many types of sequencing. In the single solution space, and we entered this space pretty early, and therefore we have built a slew of tools in order to look at single cell gene expression, TCR, BCR. We have moved into epigenetics, CytSeq, and some of the new technologies that have come up in the field. And then we use both the 10x platform as well as the Takara platform in order to do single cell sequencing. And then we have built a pipeline of tools to analyze the gene expression and even to look at various types of changes that happen when cells are responding to certain stimuli. So I know all of you are familiar with the 10x GEM technology, but I'll just give a very brief overview. So the 10x GEM technology is based on uniquely barcoded gel beads, which are shown on the left, and they're color coded. Each bead has a unique barcode. Now, the barcoded beads are mixed with cells, enzymes, and then oil, so that each cell is encapsulated in a oil bubble, and that's what is called a gem. So inside the gem, the barcoded beads actually and the enzymes and the all the other reverse transcriptases, poly-DT, all of these actually generate cDNA inside the beads. And the important thing is that each cDNA from each bead is labeled with a unique barcode. So you can actually pool this and sequence them, and then later on deconvolute these to identify the specific functional state of each cell. Now, this requires, of course, single cell suspension or nuclei, and then you can encapsulate a certain number of cells. And then because of the presence of a very large number of barcodes, it allows massive multiplexing. And then 10x has come up with many different types of applications, which we also provide at MedGenome. And you can see that we can do RNA expression. We also do feature barcoding like CytSeq, multi-seq, cell hashing. And then we provide a BCR and TCR sequencing service, ATAC-seq to look at the epigenetics. And then we are still kind of building this area of spatial transcriptomics using VCM from the 10x platform. Now, so in the 10x platform, the top part of the process I have already described. Now, in order to sequence the TCR or BCR, very specific TCR, BCR or primers unique to the sequence of TCRs and BCRs are used. And as you know that the C-terminal end of TCR and BCR has the constant region, and therefore you can design primers and do a nested PCR in order to amplify these sequences, and then do a sequencing, and then you can analyze the data. And because of this, of the sequencing, the cost of sequencing and the ability to sequence, parallelly a huge number of cells, there has been a major thrust in different applications of TCR. One is in the area of therapeutics, for example, in identifying TCRs that actually recognize very specific antigens, and therefore, and then using these TCRs as therapy. Using a TCR engineered T cells. In the diagnostics, one can look at TCR clonality in different types of disease scenario, and therefore one can monitor how the disease process is, progressing. And then even in the treatment area of treatment, for example, in vaccine treatment, one can actually do a immune monitoring by assaying the TCR and the BCR repertoires, and just to see how the treatment is affecting the individual. And then finally, this is just a cartoon to show that in the last few years, TCR-based therapies to treat cancer has evolved because of our ability to isolate circulating T cells or tumor infiltrating T cells, and then take them through either bulk TCR sequencing or single cell sequencing in order to identify TCRs that are very specific to a target, which can be a mutant protein, which can be a foreign antigen. And then these TCRs then become your therapies, and especially in human cancers. There are many companies that have come up with tools in order to exploit this particular technology. So now let me take you through the OncoPEP platform technology. So we have, as I mentioned, that we have used and leveraged some of the tools that I described in the earlier section in order to build this technology. So this platform has three components. So again, the goal of the platform is to identify epitopes that are recognized by T cells, validate these epitopes through assays, and then use this assay or the endpoint of the assay to do different types of analysis. So our epitope prediction involves a very sophisticated data-driven artificial intelligence based approach of identifying epitopes, and I'll give you a little bit of show you some data on what we have built. And the critical component of this prediction is that we have been able to reduce the reduce the false positive rates, and also identify strong T cell activating epitopes. Then, in order to validate our prediction, we have also built activation assays, both using the PBMCs or using a synthetic antigen presenting cell. And then finally, using the output of the assay, we can take the cells and take them through a variety of different endpoint. I call it an endpoint analysis. We can do single cell sequencing, gene expression, and so on. And I'll show you how we have used all these technologies and found interesting aspects from SARS-CoV-2. So the epitope discovery, or the T cell epitope discovery, is very challenging, and I'm sure that many of you are familiar with the Parker Institute and the Parker Alliance, and they published a paper in October 29th issue of Cell, and we were also a part of that study where we and others have used our own algorithms to predict epitopes. And why is this challenging is listed. The epitopes, T cell epitopes, are small peptides, and therefore the epitope space is very large. And then in order for an epitope to be recognized by T cells, it has to pass through many intracellular steps, and they are not that easy to model. And then the third point is that only a small subset of these epitopes or peptides that are presented on the surface of cells are actually recognized by T cells. So therefore, good T cell epitopes are not only candidates for vaccines, which are so elemental and fundamental in treating infectious diseases, but they are also targets for TCR therapies. So identifying good epitopes is key. So this is a cartoon showing how an T cell epitope is generated inside a cell and what it takes for an epitope to be recognized by a T cell. So as you can see, these processes are happening in three different compartments. The cytoplasm, in the cytoplasm, the proteins or antigens are degraded by the proteasome, processed, and these peptides are enter into the endoplasmic reticulum where they actually bind HLA. So I am focusing on class 1 presentation. Class 2 presentation is slightly different and also follows multiple steps. Now these HLA peptide complex then passes through Golgi and then they are presented on the surface. Now once they are presented on the surface, then these peptide HLA complexes are then screened by T cells using their T cell receptors. And only a subset of T cells or subset of epitopes will be recognized by T cells. So there are two parts then. One part is where the peptide is loaded onto HLA, and then a part where the TCR is recognizing the HLA peptide complex. Now, over the years, many algorithms have been developed to predict peptide immunogenicity. They have been combined in order to make the prediction better and better. There are programs that will predict whether a peptide will be cleaved by proteasomes and therefore be released as a small peptide. There are programs that will predict whether these peptides can bind to the TAP transporters and then they can enter the endoplasmic reticulum. But I think the most important algorithm that has actually driven the field forward is the HLA binding affinity, where the peptide is actually binding to HLA. Now, HLA binding affinity in the very beginning, it was developed by looking at eluted peptides, or the peptides that are present in the cell surface. They were eluted sequence, and then by comparing the peptides that are presented, the scientists were able to identify amino acid residues that are required for HLA binding, and these are called anchor residues. And so using those anchor residues, the HLA binding affinity algorithm was built. And then later on, additional algorithms which measures HLA binding stability, or how long a peptide is going to be present on the HLA, or what is the affinity difference between a wild type peptide and a mutant peptide in case of cancer. Some of these new tools have come which can be combined with the HLA binding affinity. So this is a very important tool that we all use in this particular area. And then the last point is that in the last 3-4 years, how foreign is a peptide also is used as a criteria for predicting the immunogenicity. Now, in spite of doing all this and making these new algorithms, the predictions are limited because it has a high false positive rate. And the reason is, as I explained, that all the peptides that are presented, they may not be recognized by T cells. And all the algorithms that currently people use, they are actually predicting the probability that a peptide will be presented on the surface of an antigen presenting cell. So we have built our prediction technology based on the TCR and the peptide MHC complex. So we used a deep neural network, and we have our model can accept different length of peptides, different features which are associated with the HLA and the HLA sequence. And then because of using neural net, we have been able to reduce the overfitting of the data. And then we found that we have improved performance compared to NetMHC PAN4. And then we have validated this on some of our own data sets and also external data set. And we have this algorithm is patented. So this is just summarizing that why we see that our prediction algorithm is superior to HLA binding. So if you just look at the HLA binding algorithm and just focus on the positive, true positive and false positive peptides. So these peptides are are mostly viral peptides, and their immunogenicity has been assessed and they have been deposited in IDB. So we have done a curation and identified the peptides that are true positive and false positives. And then when we apply the HLA binding affinity rank, we find that HLA binding cannot separate between this true positive, which is peptides that activate T cells, versus false positive, the ones that do not activate T cells. Now on the right, you can see TCR binding, and this is our TCR binding prediction score. And you can see that the true positives and false positives can be distinguished based on the score. So if you give a score cutoff of around 4, you can see that you can eliminate most of the false positive. That's one point. The second point is that our TCR binding algorithm can actually identify a lot of true negatives, and that reduces the false positive rate. So we have applied this algorithm on all the cancer mutations available from the TCGA data, and you can see this algorithm is very selective. So after passing them through all the filters, we can find very few epitopes that needs to be validated. So there are 361 HLA peptide pair that we predict to be immunogenic. Now, what is important in using this algorithm is that you can reduce the search space for identifying true immunogenic peptides. So you don't have to do a lot of assays or a screen in order to find good epitopes. So in order to validate our predictions, this is a PBMC assay. I'm sure that many of you actually use them. It has its own advantages. It's very good for screening patient samples where the number of PBMCs, the quality of PBMCs are not that great. It's a robust, high throughput, and very cost effective, and it has a first turnaround time. But one of the caveat of this method is that natural processing is not really assessed by this method, because you're adding the peptides from outside. And we have used many donors, but just to show you the data that we have used a model antigen in triplicates at different time points, just to see and optimize the process. So so we have optimized this assay so that it performs very robustly. Then, in addition, we have also built a synthetic antigen presentation platform where we express HLA and we express co-receptors. And then we also expressed our antigen as a mini gene. And then these can then be used to interrogate T cells. So one advantage of this method is that it assesses the immunogenicity of a HLA peptide pair. Here, the natural processing of the peptide is also taken care of, because you're expressing the peptide within the cell. And it is also, we have built it in a way that it is robust and high throughput and scalable. And again, using a model antigen on many donors, we have seen that our platform actually performs very well. Now we have looked at some of the most prevalent mutations in oncogenes, which were all predicted to be negative because their HLA binding affinity is very poor, very low, very weak. But we found that our TCR binding prediction was found to be positive. So when we use that in our synthetic antigen presentation platform, we found that all these antigens had a very strong immune response. So which means that when you are using the HLA binding affinity alone in order to select your peptides, you may actually remove a lot of peptides, a lot of antigens that are truly immunogenic. So therefore, that also gives a reason why additional algorithms such as our TCR binding algorithm can give an advantage. So now, as I mentioned, that now we have used our prediction tool and our assays in order to analyze, the SARS-CoV-2 and how immune responses are generated by the virus. So this is the current situation. You can see that it's all red and the infection. So this is the global infection or the infected individual count across the globe. And we have over 50 million global cases, over 1 million death. Even the death toll in the US is rising, the cases in US is rising. And most of the deaths are caused because there is the immune response is somehow goes imbalanced or becomes imbalanced, and also because of the respiratory distress, partly by the virus and partly by the hyperactive immune response. The good news is that the 2 RNA vaccines, at least recent papers suggest that they are showing a fantastic efficacy. So we hope to have these vaccines available at some point of time. One important point to note is that most of the targeted vaccines as well as antibodies, they are all targeted against the spike antigen. So we are not really targeting other proteins in the virus, and there is a reason why spike antigen is a good target to start with. Now, if you look at all the papers that have been published in the last 8 months, and you look at the immune responses that people are seeing in different donors, you will see a variety of different range of responses. One response that comes up quite frequently in multiple studies is that the T cell response is more skewed towards a CD4 and a Th2 type, and which sometimes causes an imbalance in our immune homeostasis. The CD8 T cell response is generally weaker, even in convalescent patients. When you screen them with peptide pools or with individual peptides, you see a robust CD4 response, but a weaker CD8 T cell response. The other common phenomenon that people have described is the leukopenia, that there is a massive reduction in T cells and some of the immune cell types. And then people have found exhausted T cells, a blunted interferon response, activated NK cells associated with severe disease phenotype. And then, of course, the dysregulation of the myeloid compartment. And a very recent paper also showed that the size of the naive T cell compartment is also important in this protection. And that could be one reason why older individuals are more susceptible to infection and not being able to protect themselves efficiently. And based on our knowledge of respiratory viruses, we see that a very early wave of strong CD8 T cell response, if you can generate that actually provides protective immunity, and both short term as well as long term. So finding a strong CD8 T cell epitopes, we thought when we started this program would be a key in understanding how we are fighting SARS-CoV-2 infection. Now, just to take a step back and see what is the dynamics of T cells in the case of acute infection. So a viral infection would be an acute infection. And as you can see that, so from the progenitor cells, from the naive cells, as the cells experience antigen, they are converted into effector T cells, which are the cells that are fighting the infection, and then they quickly become memory T cells. This is when you have an acute infection, because it clears the infection, so the antigen load dramatically decreases, and then it goes into a memory phase in order to protect us from a future infection. Now, in a chronic infection, you get this state where, again, you see an activation of an effector phenotype, but then this effector phenotype then goes into an exhausted phenotype, because of the chronic presence of the antigen. And this is what happens in cancer. So this modeling the fate of T cells in an antigen induced activation assay is a very critical understanding, and it is a very critical framework for our understanding of how a particular T cell might behave in the presence of a very specific epitope or an antigen. And that's what we have tried to look into through our technology. So in order to discover immunodominant T cell epitopes, we also focused on spike protein at the beginning, and I'm going to show you data on spike protein or peptides derived from spike protein. And we have taken unexposed donors, PBMCs, and then we have used our own prioritized peptides that we have prioritized using OncoPep-Vac. And in order to generate both CD8 and CD4, we have used a 15-mer peptide, but within the 15-mer peptide, we have epitopes for both CD8 and CD4 T cell activation. And then we have taken the JPT, the peptide mix, the 15-mer overlapping peptides. And we looked at the kinetics and the magnitude of the response, and also I'll show you some of the T cell dynamics that we have found through our own experiment. So, so we took the SARS-CoV-2 proteome and passed it through our algorithm, and these are the number of peptides that we got, and you can see that the spike peptide from the spike antigen we got about 36 peptides, and these are against 22 HLAs, which will cover more than 95% of the population. And if you look at the peptides, how they're distributed in the spike antigen in this cartoon, you will see that most of the peptides that we have selected. So we have not used the 36 peptides, but we have made further prioritization and selected 11 peptides. And these 11 peptides are localized in loops and in structures which are more exposed. And we specifically, used or made that selection in order to, help that these peptides could be processed and maybe available for presentation more readily than some of the other peptides that are more internal. And then, if you look at some of our peptides, you will find that they are restricted to a large fraction of HLAs that will cover the global population. Some of them also have contact residues for ACE2, which is the receptor that the virus uses to enter the cells. And then some of the peptides also have epitopes for neutralizing antibodies, but these antibodies epitopes are from SARS-CoV. So now we took the donor PBMCs and then looked at how are our peptide selected peptides, how is it behaving in comparison to spike S1 and S2 peptide pools. Now these pools have very large number of peptides, almost like over 150 peptides. Our peptide mix is only 11 peptides. So this is a 48-hour assay. On the y-axis, you can see we are looking at CD8 T cells expressing interferon gamma by FACS. We also look at 4-1BB and some of the other markers of T cell activation. And we are looking at both CD8 and CD4. And as you can see, at 48 hours, you see a robust activation of CD8 T cells by our peptide pool. And so in some of the donors, you can basically see a greater than 1% or higher activation of T cells. With spike 1 and spike 2, the activation was weaker. We can see activation, but the activation is weaker. Now, we see the same thing in CD4, that the peptides that we have prioritized are also activating the CD4 T cells. And in 7 days, we see this activation increasing further. You can see that the scale is different from here. But then when we look at the CD4, we see that the spike 2 is also activating CD4, but at a later time point. And people have actually shown that spike 1 and spike 2 can activate both CD8 and CD4, but the robustness of response is more skewed towards the CD4 T cells. So then we wanted to determine if there are how many of the epitopes that we have selected are actually qualify as immunodominant epitopes. So on the top, you can see these are again, we are looking at CD8 T cells expressing interferon gamma, and we have screened them on many donors. And the ones where I have an arrow are the donors where we have screened the individual peptides. So for example, and this is an example of one of the donors, and you can see the two peptides, peptide 3 and peptide 7, has a very strong activation of CD8 T cells, starting at 48 hours, increasing, and then coming down. And the pattern is very similar, except that with peptide 7, you see a maximal activation on 7-day. If you look at donor D167, which is another donor, you can see that multiple peptides, and some of them are truly immunodominant because they have a very strong activation, again activating the CD8 or interferon gamma production by CD8 T cells. So basically, in our screen, we find that the 11 peptides that we have selected, 7 of the 11 peptides can activate or can induce activation of T cells in multiple donors. So then the question was, and these donors, as you will remember, they are unexposed. So they have been there, PBMCs were collected 2 years before even our pandemic started, 2 to 3 years. So the question is that why are these individuals responding to a SARS-CoV-2 peptide? So several papers in the last few months have actually reported this. So this is not new, that there is a pre-existing immunity that against SARS-CoV-2 in unexposed donors. However, the hypothesis from all these articles has been that these pre-existing immunity is coming from CD4 and CD8 T cells that have seen coronavirus or common cold-causing coronaviruses. And some of the peptides which are, some of the peptides from the spike antigen have identity or similarity to the common cold-causing coronaviruses, and therefore these peptides from SARS-CoV-2 are actually activating the T cells. And so the interesting thing was that when we looked at our peptides, we find that very few of our peptides have such cross-reactive homology with other coronaviruses. All the peptides have homology to SARS-CoV, but these individuals are not exposed to SARS-CoV. But then if you look at the other coronaviruses, only a few peptides will qualify as being cross-reactive. But as I told you that 7 out of the 11 peptides have shown activation in multiple donors. So the question is then, what is the mechanism? What are these peptides seeing in this unexposed donors? So in order to address that, as I mentioned, that we did bulk TCR sequencing, and then we looked at what type of TCRs are getting amplified in our in vitro activation assay. And what was surprising, and we see it in multiple donors, that we actually see expansion of TCRs or CDR3s, which are common or which are against common viruses that we all are exposed, such as CMV, influenza, flu, or flu, CMV, etc. And so this was something new, because and this goes a little bit against the dogma that you have to have some homology with peptides in order for cross-reactive TCRs to work. But as I showed you that these peptides like PEP7 has no homology to any of the coronaviruses except SARS-CoV. So then we thought, in order to determine the specificity of response, we went and looked into, we did a single cell sequencing, and this is just data from one donor, in order to understand that are the T cells that are specific to, or not specific, but are the CDR3s that have been annotated as being specific to a viral antigen, common viral antigen, are these really getting activated and making interferon gamma? Because if you do it in a tube, then you really don't know what is getting activated. So as you can see that with PEP7, and here in the first column, you're looking at the type of cells. And as you can see in the red one are all T cells. Then you can see that the interferon gamma produced by PEP7 is huge, because all the T cells are making interferon gamma. And we see a very large expression of interferon gamma by FACS. And then when you look at the clonotype 2 is actually a CMV specific clonotype. And you can see that the clonotype 2 is expanded and it is making interferon gamma in the presence of PEP7. And this kind of suggests that the CDR3 that is being activated by this immunodominant peptide is in fact activating T cells, that those T cells which have experienced CMV epitope. So this is something that we felt is new. So we again looked a little bit further to compare the TCR beta frequency in different treatments. And as you can see that in a PEP7 treatment, you get a lot of TCRs which are very specific to peptides that come from common viruses. And these TCRs are not amplified in any of the other treatments, even with spike 1, spike 2, and in DMS, not to that extent. So then we looked at the usage of the V and J genes, and we see that in the presence of the peptide, you can see that there is a very strong amplification of V and J gene usage and enrichment of the V and J genes, both in the mix as well as in PEP7, suggesting that the activity that we are seeing with this 11 peptide mix is mostly contributed by peptide cell. So just to summarize then, this is a cartoon of our integrated platform for epitope discovery, and we have a sequencing which is then taken into a prediction pipeline to predict the epitopes. Then we validate them through our assays, and then we use this assay output in order to look at a variety of TCR sequencing, both at single and at bulk level, in order to understand the quality of T cell response. And then from there, one can then select what would be good antigens or good peptides that one can take forward for either vaccines or for epitope engagers. And then just to conclude, I described to you our technology. I described how our platform we have used to identify novel T cell epitopes. And using this T cell epitopes, we have been able to identify a robust pre-existing immunity, and then showed that this pre-existing immunity is coming from cross-reactive TCRs that are recognizing common viral antigens. And therefore, these findings together, this kind of raises the expectation that a significant of us around the globe may have pre-existing immunity, because we are exposed to a lot of common viral antigens such as flu and CMV. And this is a paper that we have submitted to bioRxiv and then we are submitting it to other journals for review. So thank you very much for, and then I can take questions. You can send the questions and then I can reply. So I think we have another 5 minutes.
Thank you, Amit. That was a very insightful presentation. We do have questions from our audiences. I'll pose the first question, which is a strong CD8 T cell response a marker for protective immunity in all infections? If so, can the CD8 response be primed via lifestyle or pharmaceuticals prior to infection?
So for respiratory viruses, the current notion is that CD8 T cell response is a more protective than an antibody response, especially because the respiratory infection happens at the mucosal surface, and that's where CD8 T cells play a role. And vaccines are most, I think the most easy candidate that you can actually use to activate CD8 T cells. So you have to identify good CD8 T cell activating epitope, and that has been the core of my presentation, that use of proper algorithm can actually help you to at least narrow down a list of epitopes that you can then screen for T cell activation.
Thank you. Have you tested these peptides in your synthetic APC platform?
We haven't done that. We have made mini genes because we also want to see whether these peptides are processed and presented. But we have done, I have not shown the data, but we have used this peptide on convalescent patients in order to see whether the patients can recognize these epitopes, and they do. And we have shown that, and that's part of our paper, we have shown that in mild to moderate disease in symptomatic individuals who have recovered from infection, these peptides induce a stronger CD8 and CD4 T cell response compared to individuals who have severe disease symptoms.
Okay, thank you. Have you or are there any publications reported for any specific HLAs that are susceptible or having any resistance to COVID?
I think for SARS-CoV-2, I have not seen any paper which describes very specific HLAs that are protective. But for SARS-CoV, there are few papers, and there is a B HLA B HLA, and I don't remember exactly the number, the specific HLA B type, but that has been shown to be enriched in individuals who have not got infected or who have recovered without any major symptoms. So that was I think that is only one thing that I can recall, but yeah, I can't be sure if new data has come out on protective HLAs.
Okay, thank you. And I just want to reiterate again, audiences, please type in your questions or even email us. One of us will be able to address any of your questions. I think we are running short of time here. With this, I thank Dr. Amitabh Chaudhary for a wonderful presentation. And I also thank all the audiences who have registered who have attended this webinar. Stay safe and be protected. Thank you, everyone.
Thank you.